Welcome. I'm Adrian Lee. I'm the professor at the University of Pittsburgh, and I'll be talking today about optical mapping to resolve complex DNA rearrangements in breast cancer. These are my disclosures. None of them are relevant to the presentation I'll be giving today. So my lab is interested in understanding the genomics of breast cancer, and through the Cancer Genome Atlas, we know quite a lot now about the genomics of breast cancer and other cancers. This is from Nature Genetics 2013, where the TCGA group separated cancers into whether they are the M class, i.e. they are dominated by base pair mutations, or they're the C class, they're dominated by copy number aberrations. And as you can see here, in the M class, you have kidney cancer, colorectal cancer, glioblastoma, lung. But my interest is in breast cancer. And you can see here, this is actually dominated in the C class by copy number aberrations, similar to ovarian cancer. There aren't that many base pair mutations. And these copy number aberrations and DNA structural aberrations have been inherently much harder to detect than these M-class base pair mutations. So here's an example of the complexity that we see in breast cancer. This is genome rearrangements in the MCF7 breast cancer cell. This is the most commonly used cell line in breast cancer. And as you can see, this is spectral karyotyping, and it shows that this cell line is near triploid, and there are translocations pretty much in nearly every single chromosome. You can see all these different colors. And this highlights the tremendous complexity of the genomic rearrangements across this cell line. I was fortunate enough to be with a group that was the first to develop methods to do this by next generation sequencing. So this was an early mate pair sequencing technique that gave base pair resolution of all of these structural rearrangements. And you can see this here. This is a circus plot with chromosomes around the outside. You can see these interchromosomal rearrangements, and every chromosome is rearranged. And it's amazing this cell line can actually grow in culture given the complexity of how the DNA is intertwined in this cell line. Now, many have actually then translated these DNA structural rearrangements into understanding the biology of breast cancer. And I just highlight one paper that actually came out this year where they looked at DNA copy number motifs, i.e. how each of those DNA copy number structures look, and found that they were strong and independent predictors of survival in breast cancer. So here's just an example from one of the figures, 3 different types of rearrangements they see. And these 3 types of rearrangements have been reported before. This one has been called simplex because you see here, there aren't many DNA copy number rearrangements in this genome of this breast cancer. Here you have complex, where you have now these rearrangements that copy number amplifications, deletions that seem to be clustered on certain chromosomes. And here you see what's called sawtooth, where basically, like that MCF7 breast cancer cell line I just showed you, they have copy number rearrangements across the whole of the genome. This tends to be the most aggressive cancers, like triple negative breast cancer, and this tends to be the more non-aggressive, like the luminal estrogen receptor positive breast cancers. Importantly, these different types of rearrangements have prognostic importance. So they calculated a classifier for these different types of rearrangements, scored those, and you can see there are some that have low risk when you have this score and have good outcome breast cancers, and then there are some that then have high score and then high risk. This is done in 2 different large data sets. Now, not only do these structural rearrangements have prognostic significance, they also potentially have therapeutic value. We know some of them already have therapeutic value. The HER2 amplification, which is one of the determinants of breast cancer, one of the major features, is obviously therapeutically targetable with anti-HER2 antibodies such as Herceptin. We have been performing structural rearrangement analysis in breast cancer and found other potentially therapeutically important structural rearrangements. And here's an example. This is a primary breast cancer where we did mate pair sequencing. You can see this is from an estrogen receptor positive luminal breast cancer. Not many structural rearrangements in this breast cancer. But when this recurred in the lymph node, this is about 5 years later, you can see now there are many more structural rearrangements. And importantly, one of them here from chromosome 5 to chromosome 6 is actually in the estrogen receptor itself, one of our main targets in estrogen receptor positive breast cancer. What happens in this patient at this specific location is the estrogen receptor, which is here, structurally represented here, gets cleaved halfway down and gets fused to this gene called DAB2, which at the time we found this, not much was known about this gene. And you end up with this fusion gene here, half of the estrogen receptor and half of this DAB2 protein. This creates, as the title suggests, a hyperactive ligand-independent estrogen receptor, and I show you this here. This is a reporter activity for estrogen receptor activity. When you have wild-type normal estrogen receptor, estrogen increases the activity. Anti-estrogens, which are important in the treatment of breast cancer, block that. So this is a classic response. This is that fusion from this patient. You can see, first off, it's hyperactive. It's much more active, 25 times more active than regular estrogen receptor. And then when you give hormone, it's completely ligand independent. So this patient was being treated with hormone therapy, yet there was obviously going to be no response because the version of estrogen receptor they have, this fusion, is completely unresponsive to hormone. And probably this played a dominant role in the progression and unfortunate death of this patient. These, as I said right at the start, these are very hard to detect, these fusions. The technologies to detect them are still challenging, but we've now detected many of them, and others. The red ones are from our group, the black ones from others' group. And you can see these estrogen receptor fusions are recurrent, particularly in endocrine-resistant, advanced, and metastatic breast cancers. So I'll show you what we've done now, moving towards another form of breast cancer, and how we've used optical mapping to try and understand a unique subtype of breast cancer. So this is invasive lobular breast cancer, ILC. It represents a subtype. About 10 to 15% of all invasive breast cancers are invasive lobular breast cancer. If it's considered an independent cancer alone, it's the 6th most common cancer type of women, with about 26 to 39 new diagnoses a year in the US. And what we show here is the unique features, unique clinical features, unique metastatic features, and unique biology of this invasive lobular cancer, this unique subtype. Its pathognomonic feature is mutation of ecadherin. So about 58% of ILC have a biallelic mutations in CDH1, generally a loss of heterozygosity and a point mutation on the other allele, causing loss. And the other forms of ILC have some other methylation and other forms for loss of ecadherin. So nearly all of them have loss of ecadherin, generally through mutation. So despite this being a very unique genetic form or a unique genomics of breast cancer, it's treated nearly identical to invasive ductal cancer, which is the major form. And there isn't really much research into this form of breast cancer, particularly because there are very few models to actually study it in the laboratory. And our lab has a strong focus trying to understand this. So as I said, the unique feature of ILC is the mutation and loss of ecadherin, which is present in nearly all ILCs. There are only 8 published cell lines for use, of which only 2 to 3 are actually publicly available. And only 2 of these have been characterized by DepMap. There are, however, additional ILC-like cell lines that phenocopy ILC, but actually aren't called ILC. They're annotated as invasive ductal cancer, or IDC. And we believe there are a critical need for additional ILC models and have many studies underway to do this. And I'll explain how we're using BioNano optical mapping for that. So here's an example of these ILC-like cell lines. So this is the ILC cell lines. As I told you, there are 8 of them. This is the ILC-like cell lines that phenocopy the ILC. And this is an example of how we show that phenocopying. So this is ecadherin here in MCF7 cells, that cell line I showed you right at the start. That's an invasive ductal cancer cell line. It has ecadherin. The cells have adherence junctions and bind tightly together because of the ecadherin. This is an ILC cell line, MM134, right here. It lacks ecadherin. That's why it's ILC. And because of that, the cells now don't attach to each other. And you can see there are individual cells here because of the lack of ecadherin. And that's the phenocopy and genotypic arrangement for ILC. This is a cell line we made from an organoid, from an ILC patient. You can see it lacks ecadherin and the cells look the same. But this cell line, ZR75-30, which is here, is a cell line which you can get from ATCC. It's labeled as IDC, yet you can see it lacks ecadherin and it phenocopies basically ILC. And we're very interested to understand, well, are these cells actually invasive ductal cancer, IDC, or actually are they actually genetically ILC? So what we're doing is we've taken all these cell lines and we're doing DNA analysis, RNA analysis, protein analysis, and other. And I'll show you what we've done with optical mapping, which already has told us some very unique features of these and exciting features of these ILC-like cell lines. So this is optical mapping for the first 9 cell lines. You can see, just like the MCF7s, they all have highly rearranged genomes. They all have unique features. Some of them have very few rearrangements, as the MM134 cell line, and some of them have pretty much the whole genome rearranged, just like the MCF7 cell line. And you can start already to see patterns of different arrangements in these cells that may relate to their aggressiveness or other kind of phenotypic features, which we're trying to relate to now. When we look at copy number data, you can see the copy number data for SNP array, which we already had on these cell lines, looks nearly identical to the data we've now got from the optical mapping. So this is an average of the copy number across all the cell lines. And you can see from SNP array data and BioNano, it looks nearly identical. So it's good. The BioNano data has given us something that looks very similar to tried and tested technologies. We find major DNA structural rearrangements called chromothrypsis. So this is looking at chromosome 17 here, and then chromosome 11 down here. And you can see through these mapping that optical mapping gets you, you can see lots of these blocks of DNA have been rearranged together. And obviously the genes within those have been totally scrambled and totally rearranged. And this is an example here where this gene here and this gene here have been turned around, reduplicated, put back together again. And you've basically scrambled not only the genome, but the transcriptome, and then ultimately the proteins that are getting made with different fusions and different gene expression. So what happens in these ILC-like cell lines when you do this to ecadherin? This is the key question. So this looks at the copy number status for ecadherin or CDH1 in the ILC cell lines and the ILC-like. You can see there's a number that have copy number loss in the ILC-like. And you can now start to see very unique features of the CDH1 locus in those what are ILC-like, labeled as IDC, but we think they're ILC-like cell lines. So this is one, HCC2218. You can buy this from ATCC. It's labeled as an IDC cell line. And when we get optical mapping data on the CDH1 locus in that cell line, it's very interesting. It has copy number loss on one allele, and on the other allele, it has a very specific deletion right around the CDH1 gene. So you see this, just this singular gene deletion here that's given to you and very clearly resolved by this bionanomap. So ultimately, this cell line has basically biallelic deletion of ecadherin. This is an event that is incredibly rare in invasive ductal cancer and suggests that this cell line is more than likely came from an ILC and is probably misannotated. It is genetically likely to be ILC. And I think this is the power of the optical mapping. It gives you this incredible resolution and mapping over long ranges of these complex rearrangements, both the chromothrypsis, but then these singular genetic deletions. This is another one. This is also SKBR3, which is labeled at ATCC as an ITC cell line, but it phenocopies ILC and looks ILC-like. When you look, this again has one allele is lost, and on the second allele, similar to the cell line I showed you previously, it doesn't delete CDH1, it actually deletes CDH3, the gene that's next to it. This is a similar gene to CDH1, and that deletion actually comes halfway through CDH1, fuses it to this gene down here, and this becomes non-functional. So basically, similar, it has biallelic disruption of CDH1, similar to the previous cell line. So to summarize, these DNA structural aberrations are common in breast cancer, and they have prognostic and therapeutic value. Optical mapping provides genome-wide information on all DNA structural aberrations in a single assay. The copy number aberration data is concordant between SNP arrays and optical mapping, telling you that it's measuring essentially the same thing. Chromothrypsis, chromoplexy, and categus are common in cell lines and tumors. I didn't show you all the data we've seen with the optical mapping, but basically you can resolve a lot of this using this large-scale mapping. And optical mapping detects complex loss of heterozygosity and structural aberrations at the CDH1 locus in both ILC and these ILC-like cell lines, suggesting that some of these ILC-like cell lines more than likely probably are true ILC cells. I would just like to acknowledge I run the laboratory with my wife. This is Steffi Oostenrijk. She's also a professor in pharmacology. We run the lab together. These are the lab personnel. We're lucky to have a number of great students. We have an incredible collaboration with our physicians, and we thank obviously the patients who've donated tissue to much of our research. And finally, thank BioNano for use of the optical mapping platform. Thank you.